what's on the inside of them. And so, what's on the inside, there are lots of things that are on the inside, but we primarily focus on those toxic related proteins that could indicate that someone is brain is changing. Their brain is slowly decaying and dying, but it hasn't reached that critical mass to where they're starting to see the behavioral consequences or the cognitive decline that most people are looking for. So I've been doing that as I said, since 2015 is when I moved to California, and I'm still doing that work, looking at blood, but I'm also expanding it into other biofluids. So blood is minimally invasive, but something like saliva is what we're considering to be non-invasive, right? If I gave everyone a cup in this room, you could easily spit into a cup with no real directions, no assistance from another nurse or practitioner, and saliva does actually seem to be a little bit more stable than blood itself. So I've been really kind of expanding my work in this space to see how early can we diagnose dementia. And again, we're trying to do this well before symptoms manifest because that's when we believe therapeutics are going to be the most the most impactful. And then the other kind of caveat, I don't even know if I really can even say this now, given this current climate, is all the biomarker work that has been done over the last 20, 30 years has frankly been done in highly educated white populations. And there are a lot of reasons for that, but nonetheless, that is not reflective of the US population. That's not reflective of the world. So, taking a step back and assessing the current methods for diagnosis, are they applicable across diverse populations is also something that I'm very, very interested in. So that was a summary of kind of like my body of work. I look at this not just in Alzheimer's disease. So the beauty about the technique is that these vesicles are everyone secretes these vesicles, and it's just depending on what marker you're looking at will determine whether you're targeting Parkinson's, Alzheimer's, frontal temporal dementia, any kind of disease that you can think of. These proteins somehow get packaged inside of them. So we actually can assess disease progression across a variety of different related dementias. So my work has kind of expanded into other areas, but the crutch is health disparities and extracellular vesicles. So if anybody has any questions for me, I'm happy to answer.
Yeah, Nahum?
Mm.
Yeah, I was really interested in how you said like you're also looking at saliva as well because it's
Mhm.
non-invasive.
Mhm.
So I was wondering like what kind of differences are there that kind of like affects your research or that you kind of have to like work around between blood and saliva when it comes to like researching these biomarkers?
Oh, very good question. So, being honest, the saliva is like the newest frontier in which I've been progressing into. So, a lot of the questions about stability, reliability, transport, do we freeze it? Do we leave it on the bench? A lot of those things are actually real still being sussed out right now. So our biggest thing is just collecting the saliva. So a lot of the large scale clinical trials that exist don't collect it. So we're really starting from the bottom. So we're now starting to get some momentum. People are starting to believe that saliva could also be used as an alternate biofluid. And so there are a few of us who are really trying to spearhead this new area. A lot of people are really, really focused on blood. And I think saliva is where blood was 10 years ago. 10 years ago, no one believed that blood could be a biomarker for neurodegeneration. And now that we're establishing proof of concept, now the field has moved forward in that direction. So pretty much saliva today is where blood was 10 years ago. So a lot of the answers to those questions we really don't know. One of the biggest component is likely going to be the contamination, right? Depending on what you ate that day, depending on your oral gut microbiome, whether there's bacteria, there's viruses, all of that potentially could be a confound, if you will. And so we're trying to assess that as well. But there's still a lot more that we have to figure out. So be on the lookout. So hopefully we'll be able to answer some of those questions. But good question.
Right, thank you.
Hmm. I don't know how it goes. I see Joanna, but I don't know the order in which it goes. So hopefully you're next.
Okay, thank you.
Yeah, Joanna, go ahead.
All right. So more about what you said about your work in addressing brain health disparities.
Mhm.
When you look at clinical blood samples, how would you separate like the genetic biomarkers of Alzheimer's disease from like the more biological changes that come from like the social economic stress that comes from these disparities, if you get what I'm
That's
saying?
Oh, absolutely.
Yeah.
That's a very, very good question. Um, and it's something, so in neuroscience, what I've learned is that people are very compartmentalized in the working that they do. So there are more prolific, more dedicated research that look at the socioeconomic determinants of health, how education, lack of food access, all of those really huge contributing factors has an impact on the biology, which then can have an impact on their biomarkers. And so, unfortunately, I am not at that stage. That's kind of like the next frontier, or what I would have said would have been the next frontier if things had not panned out the way it had once this new administration came into the fold. There were a lot of research that was halted, a lot of funds that were taken away that actually inhibited us from really asking those questions. So right now we're really at the proof of concept stage. So when anyone enrolls in a clinical trial, you get very, very basic information. So we're not even getting a genetic determinant of ancestry. We're getting self-reported. So like if you've ever taken a survey, they ask you like what category do you fall into? Like I would check the black box. Somebody may check the white box, the Asian box, Hispanic box, that kind of thing, right? So it's all self-report racial identification. So right now, that is the best that we can do is organize the data based purely off that self-identification. And then the next and probably the more deeper question that we do want to ask and that some are asking is what are some of those other confounds that actually are influencing biology? So my hope and goal is that things rebound and things get better so we can do that. But that is currently and unfortunately where the current state is. And because it's something that's already collected, that's at least our starting place, if that makes sense.
Yeah, that
Mhm.
does. Thank you for answering my question.
You're welcome.
Thank
All
you,
right,
Joanna.
Tanavi. Oh,
Yeah,
Yeah,
hi.
yeah, Tommy.
Yeah, can you see me?
yep.
So I've actually done a bit of research on blood-based biomarkers. I think it's a really interesting concept to be able to to be able to have more diagnostic methods in third world countries. One thing I wanted to get like your, I guess, professional opinion on is just you did mention like catching Alzheimer's before symptoms occur.
Mhm.
However, I've read some papers that say that a lot of people that have these blood-based biomarkers actually never develop symptoms throughout their lifetime. So how would you I guess how would you navigate that
Mhm.
the difference between like treating people before they even develop symptoms to be able to reduce their prognosis versus treating people but then provoking that fear that oh no I have Alzheimer's.
Yeah, absolutely. So I mean, that's a really good question. It's very multi-layered. So the first part has got to be education, right? And so I think you also kind of hit on the point too. So you specifically emphasize the word Alzheimer's. And so what we as researchers have to disentangle is that dementia is the broad umbrella in which we're speaking of. Alzheimer's is one version of it. And that's also something that's been very difficult in our field is because Alzheimer's is characteristic of two very specific proteins. Your brain changes in a specific way, which causes dementia. If we're talking about frontal temporal dementia, we're talking about a different protein. The end result may be the same. Dementia is still the end result, but there's a different protein, different brain regions are involved. And so the therapeutics may target one form of, say, Alzheimer's disease, but are leaving everything else unchecked. So we have to be very mindful about educating the public on everything is not Alzheimer's disease, right? And because we as researchers were treating everything as Alzheimer's disease, we were giving these cocktail therapeutics that, yes, absolutely had no impact on a small percentage of participants who enrolled in the clinical trials. And then the other thing that I really feel like the field is needing, and something that I've really been big on is multimodal interventions, multimodal diagnostics. So I also don't believe that one protein, one marker, or even one or two together is really going to encapsulate the heterogeneity of a person, right? If I'm a Black female, I'm in my 20s, I've had long COVID, right? I'm naturally going to present and I'm going to be in a different clinical trajectory than somebody who's Hispanic, who's 75, never had a virus, and maybe have had two pregnancies. Like all of these biological confounds have to go into the algorithm in order to truly predict what someone's disease progression is going to be. And so I think AI is going to actually help a lot with that as well too. The medications that people take and are not taking, a lot of these things which I think were overlooked are now starting to be recognized as potentially contributing or potentially making people resistant and protected against dementia. We've been narrowly focusing on if you have this specific gene, if you don't eat well, if you don't exercise, if it ran in your family, that was the narrative that we were telling people. But 90% of dementia is sporadic to where we actually don't know the cause of it. So I think education is really going to be key. There are some things that we do know that increases your risk, but nothing is a guarantee. So I think it requires, I'm gonna put, oh, I don't have the capacity. Can you put everybody on mute?
Yeah, let me do that really quickly.
Okay. So yeah, so I think there's a lot of things that have to happen.
I muted you too, Dr. Winston. If you could unmute, it automatically mutes you.
Gotcha. So yeah, education is going to be very, very key. We have to make sure that the correct narrative is being shared. Um, And I think that's also going to be I think it also has to be an opt-in kind of thing, right? So like we're talking about clinical trial. That's something you're intentionally signing up for. You're told the risk, you're told what you should be getting back. If we're talking about like a blanketed genetic screen or a blanketed test, like that's something that people should be educated on so they can understand like if you were to receive this result, how are you going to manage that? What does that look like for you moving forward in the future, especially if you're 20 and 30 and you know you have this risk that may not manifest until you're 60 or 70. So I think there are some cognitive behavioral holes that need to be filled, but I think we're just trying to do our best to create the test and then we'll probably figure the rest out later, if that makes sense.
Yeah, that makes sense. Of course, we need to actually be able to diagnose people before we can decide what to do with the diagnosis. Yeah.
Right, exactly. Yes. And I think we're getting there for sure. We have some really, really, really strong tests, but it's very heavily reliant on one marker, p-tau 217. And in my mind, I just feel like that just can't be the whole story. But I'm just one voice, and the people who have louder voices, they sometimes drive the narrative. So I hope that more researchers, younger researchers like myself, are really trying to answer these more nuanced questions that may be overlooked because they're a little bit more complicated, a little bit more difficult, but it's still worth asking. Mm-hmm. All right, Camila.
Hi, Dr. Winston. I
Mm,
was just wondering because I remember you said that a key part of your research was using saliva to measure the different aspects of Alzheimer's disease that you mentioned
mm-hmm,
while you were discussing your research.
mm-hmm,
I wanted to know what made you choose saliva specifically in contrast with other biofluids like cerebrospinal fluid, for example. And I
mm-hmm.
was hoping if you could maybe share anything you have planned for future works, if you're having that in mind at the moment.
Wonderful. So as I kind of mentioned a little bit earlier, when you look at the landscape of diagnostics, right, you have to weigh the pros and cons. So CSF is probably going to be the one that's the closest to being as closest to the brain as possible. However, how you acquire CSF, you have to stick a needle in between somebody's spinal cord, and you actually have to withdraw a significant amount of it in order it to really to have a meaningful impact, right? So that's invasive, and can be expensive to an extent, right? So when we're trying to think about accessibility, if we're trying to think about how can we expand this into third world countries, because America is not the only people who gets dementia, right? We're trying to save the world, not just the United States. So there needed to be a paradigm shift. So that's where blood came from. And once people jumped on the bandwagon of blood, as I said, as in everything, there are just some people who have more resources than me, have louder voices and are more influential than me, that they took a little bit of the shine of what blood can do, or what it has been able to do. So, in order for me to remain relevant, in order for me to make sure that my research program continues on, I needed to think outside of the box. And so I thought about, well, how else can we make this better? What is something that may be easy to collect, easy to handle, maybe non-invasive? And that's kind of how I settled on saliva. And this is just, we're again, at the current frontier, but people are looking at tears. People are looking at urine. People are looking at breast milk. Believe it or not, people are actually looking at feces. I don't know why, but people are having the same thought, right? Like, well, if we can do it in this biofluid, how far can we push this? Because a different biofluid is going to be relevant under different circumstances, right? And so if we're trying to really make this a diagnostic tool for all, then we need to be flexible. We need to be able to make sure that whatever we're creating is not context specific, or even if within a certain context, there's still a test that can be used, and no population, no area is going to be left out of that. So as far as what I have planned, so we're currently running some saliva assays now. As I mentioned earlier, a lot of people weren't collecting saliva. So now that we have biobanks that are collecting saliva on our behalf, we're actually running match samples to really see if what we see in blood replicates what we see in saliva. hmm and
Thank you
I do
so
not,
much.
you're welcome. Thank you. Okay. So I see a couple more, and I'm gonna try to be a little bit more short-winded because we're right at half. So, I can't pronounce your name. Ashi?
Yeah,
Yes.
Ashi.
Oh, wonderful.
Sorry,
Okay.
but wait. Sorry, I'm going to interrupt really quickly. After Amaya, we're going to pause questions for now from hand raising because we did have a Google form where you guys were able to submit questions, and those people did submit their questions earlier. So we want to get to those questions. So then everyone after Amaya, you can re-raise your hands after we get to this part. So, because Dr. Winston does have a little bit, quite a bit of responsibilities. She's a busy person.
hmm
So Ashi, you can continue. I'm so sorry for interrupting.
hmm
Yeah. So, I was just like really interested in the part about extracellular vesicles,
hmm
and it just made me wonder, do you think that like differences in those like extracellular vesicles could help explain why people with similar levels of Alzheimer's biomarkers can sometimes have like really different symptoms?
I think it's possible, right? So the reason why we look at the EVs is because they're like these durable little structures that are not easily degraded, like that you can't really break them down as they are traveling. So we believe these EVs are coming from the brain. They're somehow getting themselves into the peripheral circulation. We're pulling them out of the blood, saliva, urine, all these other places, right? So they got a long way to go. And as you can imagine, when you go on a journey, not everybody makes it. So it's very possible that the journey that these EVs are taking, and the fact that we're actually able to identify them in these different biofluids, can suggest that there is a lot happening. There's a lot of buildup that's happening in the brain, and these EVs are really trying to package and get it out as quickly as possible. So, really, what we're quantifying is how much of that toxic protein do we see, and it's at a very specific time point, right? And so the more you have of that protein is likely suggested that the more you have built up in your brain. But it's really just a window. We're just trying to assess what's happening, because obviously you can't decapitate somebody, take their brain out and put it back in, right? So it's really just giving us a proxy of what's happening. The real question that you're asking, there are hosts of other things that could help us understand why people have different symptoms, right? It could be diet, it could be sleep, it could be their biology, it could be their environment, it could just be you're a female. Like, there are a whole bunch of things that could also contribute as to why someone would have greater signs of dementia than the other. What looking at EVs is just giving us a way to be able to capture it more accurately. I think that's what my EVs are doing. Okay, go ahead,
Thank
Amaya.
you.
Mm-hmm.
Hi, Dr. Winston. So I just wanted to go back to like what you said earlier about your work on like racial disparities in Alzheimer's disease. And so I think previously I've like looked in a little bit to the amyloid precursor protein and
Mm-hmm.
how like that protein is like specifically expressed at different rates that can like affect the progression of Alzheimer's disease in someone's life. So I was just wondering if APP plays like any role across like multiple ethnic groups and how like it might cause like a difference in
Mm-hmm.
what age like Alzheimer's commonly sets in across those ethnic groups.
Yeah, absolutely. So the APP precursor protein, so it's this long protein, right? And it does a lot of basic biological functions. Like everybody has one. We absolutely need it in order to do lipid metabolism, so on and so forth, right? What happens is this protein gets cut twice, and when it gets cut, it creates amyloid beta. And that is the toxic protein that builds up. It builds up in a lot of dementias, but what it's most known for is being one of the pathological hallmarks for Alzheimer's disease, right? So there are a couple of questions, right? If everybody has this APP molecule, then how often is it getting cut, right? Some people may cut it slowly. Some people may cut it faster. Some people may not cut it at all, right? So you can look at enzymatic activity. That could be influenced again by disease, by diet, genetics, cultural influences, sleep. There are a whole bunch of other like factors that could increase your risk, right? And then now when you got the protein, then well, okay, just because you build it up doesn't mean that it has to stick. So how are your waste removal mechanisms? Is it getting washed out nightly? So if you're not sleeping well, then that's your major way to clear things out of the brain. So APP is kind of where everything starts, but there's so many things that happen in between that could cause the protein to get built up or could cause it to actually to get efficiently removed out of your system. So a lot of the questions that y'all really are asking are the reason why people are still in the business that we're in today is because the real answer is we just don't know. Like we can identify certain populations and we can say that, I mean, I'm gonna speak for myself. Like in the African American population, we have a huge genetic risk profile. For whatever reason, we are genetically predisposed at higher rates to getting Alzheimer's disease. However, we have less amyloid in our brain. Right? So when we get a PET scan and we measure how much amyloid, we actually significantly have less levels than other groups, but for whatever reason, we're at a higher risk of getting dementia. So that means there must be something else happening, right? And to an extent, we don't really know what that something else is. Is it another protein? Do we have more inflammation, right? Do we not have good lymphatic clearance? So that's what we're really trying to figure out. And that's why we need more robust and diverse clinical trials because we can't even begin to ask those questions across different racial and ethnic lines if we don't even have the participants involved in order for us to be able to ask those questions. So I hope I answered it. But the real big answer is like, honestly, we just don't know.
Yeah, you did. Thank you.
You're welcome.
Thank you guys so much for asking your questions. I'm so sorry that for those of you guys who had your questions, we're gonna have to move on just a little bit and then we'll get back to those on a later time. But one thing I did want to mention is, first of all, such an impressive and outstanding background from Dr. Winston. And you can definitely tell how, passionate she is about her work and how she's actually really, making an advancement towards research in the future. And one thing I really wanted to emphasize was research problems. So, Dr. Winston, when we were, discussing the capstone projects that the students were going to complete, we emphasized a research problem, right? A problem that they're trying to solve. Rather than just doing research for the sake of doing research, it's having an objective to solve an
Hmm.
existing problem in the real world, right? For example, in your research, that would be ethnic and health disparities, and also inaccessibility to treatment for Alzheimer's disease, or even testing for Alzheimer's disease. And so
Hmm.
I wanted you to give a little bit of emphasis on how important it is to have a research problem and focus on, actually solving something.
Absolutely. Yeah, you do have to be focused, right? So like I have a very broad goal, right? Early AD diagnosis, but you can do that in so many different ways, right? So like as I said, AI is about to be a huge component of this world. Whether I'm looking at blood, some people only do neuroimaging. So like you have to find your niche because you're not going to be able to solve this problem by yourself. But if you have a strength, if you have a very specific and narrow focus on how you're going to achieve your broad goal, your broad research question, that's how you can really, really chip away at the problem and get us just that one step closer, right? So that's why hypotheses are very important, very specific hypotheses, something that's testable. Right? Something that you really can see and get like a yes or no answer at the end of it. And if it's no, cool, then you just redo it again and come up with a new hypothesis and kind of, go back to the drawing board. But yeah, you got to be very, very specific. You can't have anything pie in the sky like, oh yeah, I'm going to solve Alzheimer's disease. That's never going to happen, at least not in my lifetime, probably not in yours either. But you can do things that can help get us one step closer. And that's where specificity comes in.
Thank you so much. So
Mm-hmm.
now we'll get to your guys' questions. You guys did have some great questions. And it's super important that you guys get this type of mentorship because it's something that, a lot of high school students lack. They're not able to really connect with professors. So, Dr. Winston, I'm going to ask you a couple questions and then you could just answer them for the students. So the first question is, how exactly does statistical analysis happen in research and what tools and methods are used?
Oh, that's a good question. Unfortunately, it's out of my that's above my pay grade here. So what you'll learn if you continue to stay in the space, like we are compartmentalized, right? Like I have my niche, I have my strength, and biostatistics is not one of them. But I collaborate with biostatisticians. They like go get PhDs. This is like what they do. So when you're working with thousands of participants and you're accounting for hundreds of variables, like that's not something a human brain can pretend can compute. So there are algorithms, there are programs that are intentionally designed to do that for us. You just have to know how to write the code in order to ask the correct question. So I'm thankful that I have collaborators and I have a staff that I give them my research question. I tell them these are the groupings and the comparisons I want to make. They write the code, they throw it in the program. Y'all are probably more familiar with it than I am. That's the R, the Pythons, and all the other things that exist. And then voila, it comes out with an answer for us. So that's how it's done from in my lab. I have a collaborator that does that for me.
Thank you. And yes,
Hmm.
Celis is amazing.
Yes. Hmm.
The next question is, are literature reviews genuinely useful to the scientific community and considered original research?
It's considered a review. It's in its own separate category. I like reviews. If they're done well, it pretty much does the work for me. Instead of me having to survey 20 papers to kind of get to one thing, I go to a review, the latest review, and it's going to kind of culminate all of that past research for me together into one document. So they're very useful for the field. If they're done well, right? Like if you're not really kind of getting into the nuances and the nitty-gritty of whatever your topic is and there are major holes that are missing, then it won't be received well. But I think they're very valuable contributions to the field. I guess they're just a lot of work in a different way, right? Because if you're doing an original article, that means you designed the experiment, you did the experiment, you interpreted the data, you wrote the paper, that kind of thing. So that could be a lot of work on one end, but a review article, you got to read a whole bunch of literature and you're trying to kind of like put together themes across a variety of different investigations. So that could just be, again, it's just a lot of work just in a different way, but they are useful if done well.
Thank you. And great question. The
Hmm.
next question is, what advice do you have for students interested in majoring and or studying in neuroscience?
Um, That's a good question. So I didn't like I have a biochemistry degree. Like when I was in college, neuroscience was not a degree. So I didn't have that as an option. I think how I fell into neuroscience, which a lot of people do like a personal connection, right? So like my uncle was diagnosed with Alzheimer's disease, but again, this was 10 years ago when we didn't have the real language to not differentiate that not all dementia is Alzheimer's disease. So given certain aspects of his trajectory, he actually probably had more frontal temporal dementia, but nonetheless, to see the progression and the decline in his health really kind of got me thinking like, okay, like I like the brain. I know that I want to do something translational. I just don't want to do something that's not really going to have a huge impact on the field. So I think I say that to say whatever your inspiration is, like make that the reason why you want to do it. Like know your why, right? Like why are you in this space? Why do you want to learn this? Whether it's personal, whether it's something that's interests you, whether you see it transforming the world in the next 10 years, 5 years, whatever, that should be the reason why you want to do it. Because if your heart's not in it, then it don't matter what you study. It's not going to be meaningful for you and you're not going to want to continue on. So I would just say, yeah, know your why, know what interests you. And if you don't, like, it's okay too to bounce around. Y'all are young enough to where, like, you can go in a cancer lab, you can go in cardiovascular lab, like, go test out a bunch of different things so you can truly figure out, like, what you will like and what you do want to do, and then go based off of that.
Awesome. And that's amazing advice, honestly. Just do what you love to do, right?
Yeah, absolutely.
The next question is, and this one is actually pretty popular. It's kind of why a lot of the students are here. When in college or even in high school, what is the best way to connect with professors for research opportunities?
Honestly, if you're not in some sort of like pipeline program that's intentionally set up to funnel students into labs, honestly, you either got to know somebody, like your mom knows somebody who work in the lab and you get hooked up that way, or you're just gonna have to like just cold call, like not cold call, but like just send blinded emails, right? I get them all the time, and the biggest problem that I have is like a lot of people are just not in the physical location of where I am. So I have a wet lab, right? So my lab requires in-person, hands-on, like, that's how I can train and mentor my students, right? But there are other labs that are a little bit more blended where doing a remote position, more biostats, more data analysis, data management is built and infiltrated into their lab. And every lab is different, right? And I choose how I want to run my lab. So I choose to have a wet lab and collaborate on the biostats part. And so that's like finding the lab that meets your needs is also going to be a little bit of shopping around. So if you see someone that has a research interest that you think may be conducive to you, then honestly, you just got to send them an email and say, hey, I admire your work. I see that you're doing this. If you're willing to take on a high school student, a college student in the summer, please let me know. And you like mentorship is a core of majority of the faculty that I know. And so if you email them, the worst they can do is say no, right? And it may could just be a logistic thing. It could be, maybe ask me next summer or ask me during spring break, and it could be a timing thing. So that to me, I would say is probably the easiest way. Like tap into your own network first, like tap into your counselors, tap into your teachers, because they may know someone. But then if you are just looking for, if you're going on student like school websites and you find someone's research that you like, honestly, just email them. Just straight up, I mean, create a very nicely worded, introduce yourself email, properly formatted email, and then just see what they say.
Awesome. Yeah, and I think that's so important. I've really emphasized networking, within this program. I think it's a big thing, not even with just students, but programs itself can really connect you with the people that you need to further in your career. And then, yes, we'll go over emailing later. That is a big thing that can get people
Mm,
research positions. One thing I will say though is don't expect your first email, your second email, even your third email to land you a research position. A lot of
yes.
professors are very busy. They might not have the room, like Dr. Winston said, it's really logistical. So sometimes they can't even have you in their lab. It might not even be a remote position. And so, you know, don't be, discouraged by a couple no's.
Yep.
So, really focus on what you want to do, and it never hurts to email.
Yeah. And email a couple times too, because like I mean, like sometimes just like I may overlook the email, right? I may have read it and then forgot to come back to it. And so I would also say like don't take that as like a slight against you. Sometimes logistically, like we get emails all the time. I literally probably get 50 to 100 emails a day for a bunch of different things. And I got to prioritize what to respond to and what not to, what to respond to. So if you send someone an email and they haven't gotten back to you, like it's okay to ping them. Now, I wouldn't keep pinging them, like maybe one ping, maybe another ping. But after that, like if they're not responding, then yeah, just move on. And then the other thing too is like sometimes you just got to get your foot in the door. So, like, say for instance, your heart is really, really set on neuroscience. However, your mom knows someone who works in a cardiovascular lab. Like I would take the position in the cardiovascular lab. And so now you can say, I have this experience. And then that actually gives you a little bit of leverage when you want to go find another experience. Because the fact that you've been in a lab, you kind of have a general understanding of how it works, more people will actually value that versus someone who's had zero experience. So keep that in mind as well too. Never really pass up an opportunity if it may not be exactly what you want, but it could literally be the stepping stone to get you what you need.
Awesome. Definitely amazing advice. You guys really, I know you guys want to do research, and this is what your stepping stone is to get a sort of hand at research. So I really take that, like strongly. The next question is, what are some key components of a good research poster?
Mm. Everybody's going to answer this differently. So I am a visual person. So I think attractive colors, bold fonts, not long paragraphs, right? Like when someone is walking past a poster, they probably gonna be there 5 minutes at the most. So you want to find something that's gonna draw the attention in. So I think visuals are very key. So like when you like for a method section, right? You don't need to list out all 20 of the steps you did 1 through 20. Have a diagram. A visual diagram so people can see the flow of what you did. Make your conclusion straight to the point, like simple sentences, very simple, high level sentences, but again, not paragraphs. So I think to me that would be my main takeaway. And then also it's a creative thing as well too. Like people design posters 25 different ways, right? So you should include your creativity into it, but also you may want to look and get inspiration from others and look to see how would you feel if you walked up on a doc on a poster that was nothing but paragraphs and words. Would you want to stand there and read that? Probably not. So always put yourself in the other shoes who's looking at it and see what would catch your attention, what would make you stop and say, hey, this looks like cool research, and then do that.
Amazing. The next question is, what makes a neuroscience capstone literature review scientifically strong and meaningful? What should they include in their reviews? And yeah, what should they include in their reviews and what makes it strong?
Well, that's gonna vary based off the topic, right? But what makes them strong is that they really gotta be encompassing, right? So like, say for example, if you were doing diagnostics for Alzheimer's disease, right? That's your broad topic. If there are five major diagnostics that everyone is using, but you only cover three of them, right? So now you're incomplete because somebody's going to say, well, what about the other two? Why didn't you talk about those? And then you always with reviews, you always want to capture what's current, but you also want to kind of speculate on what the frontier is because that's what a lot of people go to reviews as well too. Sometimes people go to reviews to steal ideas. Like what are people talking about? What is something else that could potentially be my next project? So you always want to think about the future. So you're kind of cementing a point in time. What's the current state up until the point that I published this paper? But then you should always have a section that's like future, like a real big future direction thing. Where do we see this going? What is something that's we could have talked about, but there's not enough literature on, but we see something happening. Like you got to have that future aspect. So I think that makes a good review It's got to be very all-encompassing, but you also need to project into the future.
Awesome. And we'll move on to our last question. And this one is actually, I think, a pretty interesting question. What neuroscience research, what neuroscience research besides the one that you have been working on, do you think will make a difference in our future that you've heard about?
That's a good question. Oh man. Mm. Oh, that is a good question. Do I have any insider tips? Wow. Um, well, I don't know if it's going to be the wave of the future. I have no clue. But I'm thinking about what I think is like, I didn't think about that. So looking at extracellular vessels, like I kind of pop into multiple different spaces, right? So EVs is one thing, Alzheimer's disease is another, and my research just blends the two. So when I go to the extracellular vesicle conference, they're taking EVs from plants. Um, and then using those as delivery systems, right? So kind of like how we would take a supplement to, deliver a vitamin or something that we need. It's possible that we can take these vesicles, extract them from natural healthy products, package them with things that we may need, like maybe a novel drug therapy, something that'll correct a mutation, could be a growth hormone, that could fit inside of EV, and then delivering it back to a patient and hopefully having that be a way kind of to be used as a novel therapeutic. So that's something very specific to the EV space. And I'm pretty sure something is going to come to me like as I'm driving home or something like that. But that would be the thing that kind of came to my mind.
Thank you so much. And yeah, that is
Mm-hmm.
super interesting. I love that question. I think there's so many broad topics. We really
Mm-hmm.
focused on neurodegeneration, but like you guys selected in your applications, neuroscience is such a broad encompassing, topic. There's so much in the field. And so, I've gotten a lot of DMs from you guys talking about your topics, and it's so crazy to see how you guys are taking this field and, really going narrow and specific in so many different ways.
Yeah.
So thank you guys for the questions. We'll move on from the questions for now because we do have student presenters and I didn't want to miss out on you guys. We are nearing the end of our time. So what I will say is if it is 5 p.m. and you do have to leave, you are free to leave. We did say that it was going to be a 4 to 5 meeting, but if you would like to stay longer to listen to presentations or you are presenting, you guys can stay longer, ask questions, and yeah, save questions for later.
Mm-hmm.
And then Dr. Winston, if you do have to go,
Yes, unfortunately, I have to go, but if you just let me know when the next meeting is, I'll see if I can try and join. So, and then if there are more questions people want to ask me, I can sit around for that. But just let me know offline, and then I'll try and make it.
got it. Of
All
course. Thank you so
right.
much, Dr. Winston.
Of course. Well, thank y'all so much. I appreciate all the questions. I'm sorry the ones I couldn't get into. But if there's something, I'll put my email in the chat as well too. So you can also email me and I'm happy to respond that way. So my email's in the chat. All right, y'all. Thank you.
Bye, Dr. Winston.
Thank you so much.
Awesome. So unfortunately, Dr. Winston did have to leave. Her email is in the chat. What I will say is before you guys immediately go and email her, really think about your question again. Like she mentioned, she gets hundreds of emails a day, like around that much. And so we do have a lot of participants in um, obviously in this program. And so we don't want to bombard her with emails, but she did give you guys permission, so take that opportunity. And there's only a couple of you guys in here. There's not so many. So if you guys want to email her about her research, go ahead and ask her questions. Um, and now I'm so sorry. I know that we ran behind schedule. We had a lot of questions, but we do have some student presenters. We'll be moving on to our research introduction presenters. So Tanvi, if you are here and you would like to present.
Hello. So, I'd like to just present the independent research project that I've been doing. So, there's actually a major problem. So, firefighters, soldiers, and sometimes even ordinary people must make fast, act or hold back, run or stay still decisions. These are known as inhibitory control. And the part of the brain that actually controls for these inhibitory control decisions, this type of cognitive function degrades during the heat. Um, however, while this evidence is widely supported that heat does degrade this cognitive function, the the evidence is largely behavioral. It only focuses on the outcome, not the actual cognitive processes behind it. So we still don't know how the brain's inhibitory control system behaves under repeated, prolonged heat exposure and exercise, whether it adapts, and also the cognitive mechanisms behind it. So the main objective of my research study is to measure how do individuals vary in their frontal EEG signature of inhibition across repeated bouts of exercise in the heat. So if anyone has any
Awesome.
questions, feel free to ask.
That was a great, I think, research problem. Um, and I'll ask that we have questions in the chat, and then Tanvi, you can get to them in the chat because we have to, I'm so sorry, we have like really limited time. But our next presenter will be Arnav.
Hey guys, my name's Arnav. I'm from Illinois, and my project is on Alzheimer's disease and potentially finding a way to prevent it using a new method. So here's my problem. Alzheimer's disease is the leading cause of dementia, characterized by progressive memory loss, confusion, and eventual loss of independence. It accounts for 60 to 70% of the estimated 55 million dementia cases worldwide. Biologically, this disease is defined by two hallmark features in the brain. The first is amyloid beta plaques, which we were talking about earlier in this meeting. And the second is tau tangles, twisted protein fibers that form within neurons. Combined, these two features are considered the defining pathological markers of Alzheimer's disease. The conventional assumption has been that these plaques and tangles are directly responsible for cognitive symptoms like dementia. However, research has identified a striking exception. Some individuals, upon autopsies, are found to have plaque and tangle pathology comparable to that of an Alzheimer's patient, yet during their lifetime, they exhibited no cognitive symptoms whatsoever. There's a certain name for these types of individuals. They're classified as NDAN, or non-demented with Alzheimer's neuropathology. It's basically just asymptomatic Alzheimer's. This finding is significant because it challenges the assumed relationship between pathology and symptoms. It suggests that there is some biological factor that is helping preserve cognitive function and would help with patients with patients with Alzheimer's disease. This phenomenon I was just talking about is referred to as cognitive resilience. So why does this even matter? The objective of this study is to identify the mechanism responsible for the protective effect in NDAN individuals. So understanding this mechanism could provide a novel target for intervention, and instead of just focusing on like plaques and tangles that are directly causing Alzheimer's, this research aims to characterize and potentially replicate the natural processes that allow NDAN brains to remain resilient with the focus in dendritic geometry. Thank you, and yep, that's what I've been working on.
Thanks so much, Arnav. I think that's a great project. And again, it's related to Alzheimer's disease, so I think we already talked about how that is a research problem. I am going to stop the research introductions for now and get into our summaries because I think that for the video, some of these questions here are going to be really helpful. And so what I'm going to ask is that for those who were selected to summarize and read their summaries, instead of reading their summaries, they just answer the three questions. What was easy? Sorry. What tool? Or sorry, there's four questions. Oh my gosh. What tool helped you the most? What was easy to understand? What was confusing? And why is your summer good? Or maybe why not if you think it isn't that great. So our first participant would be Camilla. If you're still here.
Hi guys, my name's Camilla. So for the first question, what tool helped guide you most in your summary? I would say that the two main tools that guided me most were consensus, which helped me kind of like find similar articles to the article we were reading about like the infacing smiles when I needed to kind of like reference other literature if I had questions about a certain topic that they mentioned, or like if I needed to know what something meant. I used that to kind of cross-reference with other articles and figure out questions I had as I was reading. And then another tool that I found very useful was PubMed. I kind of used it for the same reason, but I found that I had like a broader scope of literature to sub-reference while I was reading with that one. What was easy to understand? I thought in terms of like reading the article, I would say that I found the methodology section most easy to understand. I felt that they made that like very clear, even though there were like obviously you can't know everything the first time you read an article, but I felt that they made the experiment they did very clear, and I thought that helped a lot when I was trying to write about research objectives and compare their study with their results. What was confusing, I felt that the discussion section was a little bit difficult to interpret at times, just because there was like a large scope of different topics that they mentioned in the discussion section of the paper, but I didn't think that it was impossible to interpret, just like a little bit difficult at times. And then I think what might have made my summary a little bit better was if I had invested a little more time into it. For any literature review, it's very important to kind of like, as I said earlier, to read other articles relevant to what article you're already reading as like your main piece of scientific literature. And I feel that having more time to kind of compare and contrast it with other studies that cover similar subjects might have made it a little bit better.
Thanks so much, Camilla. And I think that, you know, you looking into other articles is something that's definitely really important. It helps you really understand, the broad topic and, generalization of the topic and paper that we summarized. So great work. Our next presenter would be Vihan. Again, we're running out of time. So instead of just reading out your summary, I want you to answer these 4 questions.
Okay, I'll be quick. So what helped me understand the paper, I think consensus was a pretty big part, and also like reading other like psychological articles, like the facial feedback hypothesis, and just understanding those competent like those concepts really helped me understand like the paper as a whole. And what was easy to understand, I think I've taken AP Psych in school, so I'm pretty familiar with the facial feedback hypothesis. So the concept that if you're like, if you see that there's like an avatar smiling and you also start smiling, your mood also starts changing. So I think that concept was like easy for me to grasp. And what was confusing about the paper, I think that why like them like feeling that they can control the avatar, why that makes them like happy, and like if the avatar is sad, why like they don't become sad. That like distinction like didn't really make sense to me. And yeah, the technical terms such as like ownership and agency, they were also really confusing. Yeah, and what helped make the confusing parts clearer, I think that understanding the two concepts like ownership and agency, like ownership being that the avatar feels like a part of me. So they like feel connected to the avatar and that's why their mood changes. And agency being like they're controlling the avatar. So their mood also changes. I think those two concepts made things a little bit more clear for me.
Awesome. Thanks, Vihan. Our next presenter will be Ruth Feek.
Oh, can you hear me?
Yeah, if you can just answer the 4 questions.
Okay, yeah, sorry. I'll make it quick.
You're good.
So, the tool that helped me the most is Google because it helped me like identify like unfamiliar terms, and especially like the niche technical terms here. What was easy to understand is the experiment design, the author of this art or researchers made it really clear what the aim was, the independent variables, the dependent variables, and just like the experiment design. And as I mentioned before, what was confusing was like the technical jargon, right? I think my summary did a pretty good job because it clearly outlined the claim or the variables, what they did, and results. One thing that I could improve is that it was a little bit vague. It could be more specific.
Awesome. Thanks so much. Our next presenter is, sorry, I have to, Kayla. And thank you guys for
I'll
sticking
be
around, by the way. Sorry
oh,
about that.
I'll be really quick because like basically everybody said similar thing. But I agree. I think the bibliography was especially helpful with like all the citations and reading into, I think it was like the theory of coding that they mentioned that was a little bit complicated. And I also think that the results were a bit difficult to interpret just because it was mostly about them like proving the statistical significance of the results. So yeah.
Thanks so much. Next, we have Joanna.
Yeah, so for the first question, I feel like the note taker tool with the PDF helped me a lot during the summary process. I used a lot of like the different colors with highlighting just to kind of highlight the main points, like other topics that I wanted to revisit, as well as words that I was confused about and wanted to come back to later. Um, for me, I feel like reading it multiple and multiple times made it a lot easier to understand, especially the visual diagrams, like where you can visualize the different data points, just kind of seeing their words put into something that you can see and kind of connect helped a lot. For the confusing part, I feel like the jargons is kind of a point of confusion across many research papers. So I had to search up quite a few vocabulary terms. And for my summary, I feel like the first draft that I wrote was not that good because when I put it into Consensus AI, I was having doubts about it and I didn't want to submit it yet. So I asked them to kind of give me a grade and feedback based on what I wrote. And I originally got a C plus or like a B minus. And so I went back a couple times just to incorporate Consensus's feedback and just to improve my summary. Yeah. So it would be better if my paper was less like specifically detail oriented and encompassed more points in the study. Yeah.
Thanks so much, Joanna. And I think the main reason I wanted to get to this part and actually include this within our meeting today, especially for our recording, is that everyone uses a tool in a different way, whether that be note taking, using AI as a tool, using, search engines to search up terms, because of course, like I mentioned before, you're not going to know every term in the research paper. And that's not just me, that's not just you, that's also the professors and the researchers that, they conduct reviews, they conduct their own research projects. And when they look into reviews, they all the time come across terms that they don't understand. I've firsthand worked with a bunch of professors, and I've noticed that happen a lot whenever we looked at whatever research project or research paper, there was terms that they might have not understood and they had to look them up. And that's completely normal. You're not going to know every term. And the second part was that, everyone found something confusing. And this paper wasn't, like I said, the easiest paper. It wasn't the most difficult. There's like neurobiology where they have all of this really confusing topics and protein cells, whatever it might be. But even even a research paper like this, where the topic itself might be a little bit more kind of simple to understand, where you see someone smile, you see a figure or avatar smile, and you smile, your mood might be uplifted. There's still so much terminology behind their actual research. It's not as simple as smile, smile, you're happier. Like I made it seem when we first did our summary, and I brought that topic up in our first meeting. It's actually much more complex than that, and the research behind it is so much more significant. And that's what I want you guys to focus on in your research papers or your research projects. It's not just the broad baseline topic. It's getting more narrow and specific and in depth to really, narrow down what this research topic is about, right? And kind of like what Dr. Winston said, make sure your paper, your project is really all encompassing. So it shouldn't be too broad, but whenever you're going specific, let's say your project is about um, firefighters and heat and you know the damage that has on damage and impact that has on your neurology, right? Your cognition. You have to really focus on all the aspects of that. And so we do have one last presenter, and that is for our research introduction. Sarah, if you would like to present, I'm so sorry for the wait, but you are able to present your problem. And I want you to really focus on the problem and objective portion.
Yeah, okay. Um, One sec. So basically, my research problem focuses on neurodegenerative diseases such as Parkinson's disease and ALS, which both involve a progressive neuronal loss driven by localized neuroinflammation and cellular stress. While systematic anti-inflammatory and neuroprotective compounds such as EGCG show strong therapeutic potential in cell models, such treatments rarely actually work in human clinical trials. And we know which genes and pathways cause brain cells to die in diseases like Parkinson's or ALS. And we even have tools like CRISPR that can turn on protective genes to save those cells. However, the problem lies in the fact that we do not have modular, genetically targeted delivery platforms that can selectively internalize into specific vulnerable neuronal subpopulations based on their surface transcriptomic markers. So the objective of my research project, as a literature review, is to evaluate existing research on cell surface neurotranscriptomics and targeted nanocarriers in order to propose a conceptual framework delivering CRISPR-A gene therapies selectively to dopaminergic neurons in neurodegenerative diseases like Parkinson's. Yeah.
Thanks so much, Sarah. Great topic. Again, like I said, there's so many broad topics with neuroscience. And thank you guys so much for staying. This meeting was supposed to be so much shorter. There's only like 3 slides other than the introduction. But again, the questions took a long time. And I want to actually commend all of you who stayed. That's something I keep note of, right? And we talked about leadership positions and recognitions. So your presence here, your participation within getting things submitted and asking questions, talking with the professor, interacting with your peers, and even again, just staying in the meeting and hearing out your peers is so valuable. For those of you who presented, that's such a big thing as well. And so that's something we definitely keep note of. I want to make sure that you guys know that these recognitions, we actually take it pretty seriously. Like Dr. Winston, again, she was looking at people who were participating, asking her questions. Many people weren't able to ask her questions. She put her email in the chat because she wants to see who's interacting. Again, like I said, don't email if you're not, if you're just emailing for the recognition, that's not going to do anything, right? It's more so I pick and I really look at that. But thank you guys so much for being here. It really does mean a lot. And that is all for our meeting today. So make sure you guys get caught up on your tasks, which many of you haven't submitted your research hypothesis question and keywords yet. Which if you haven't, that's okay. I just sent them out like technically today, really early in the morning. And so I want you guys to get working on that. And we'll get our next paper out very soon for you guys to work on, and just keep working on your introductions. From what I've heard from many of you and seen in my DMs, you guys are really working on that. Whether you're working on a group or working solo, you guys are progressing at a really great pace. And so that's so impressive. And yeah, that is all for our meeting. So you are free to leave. Thank you guys so much for coming. Yeah, Joanna.
Yeah, sorry to bring this up like at the end of the meeting, but one of my group mates, Shanzita, she messaged you on Slack about a research question because we wanted your thoughts on it before we submitted it. I did just send it to you in the chat. It's rather long. If you don't mind
Yeah, I
reading
see
that
it.
over, that'd be amazing. Thank you so
Yeah,
much.
I'll definitely read that over and save it. And I know I just told everyone to leave, but I will say, if I haven't gotten to your Slack message, this is also for the recording. If anyone, I mean, I don't think people really watch the whole recording, but um, if I haven't gotten to your Slack message or your email or any sort of communication, just send it again, or like kind of like what Dr. Winston, just send it again or let me know. I kid you not, maybe like an hour before the meeting, I was getting a Slack message and an email every single second. Like I kid you not. I was getting, my phone was buzzing like nonstop. Like it could have been used as like a massage gun or something. I was like, I got so many Slack notifications. I had to like take a 3-minute break. Um, and it's a lot. So if I haven't gotten to it, and I'm so sorry that I haven't gotten to it, I did see Shanzi's message. Wow, I just completely butchered that. I did see her message. I wasn't able to get to it because I have, there's a lot going on. But yeah, just send me the message again. Thanks for letting me know. I will get to that. And for the rest of you, that it's the same thing, right? If I haven't gotten to you just yet, send me another message. I'm getting so many messages. So, and then we're also working with USC to get affiliated. That was a huge thing. So yeah, and yeah, sorry for dragging this out. You guys are free to leave. Thank you guys so much. Bye bye.
Yeah. No worries. Thank you so much. All right.
Thanks. Bye
Have a
bye.
great day.
You as well. Bye bye, guys.